because both branches of immunity require helper T cells, so we'll just talk about helper T cells and cytotoxic T cells, take a break and come back and talk about the B cell response. So for the cellular immune response, we have to get helper T cells activated in order to get anything going. For both branches you do. But in order for our helper T cell to become activated, for its antigen encounter and activation, it's going to have antigen presented by our antigen presenting cell in its MHC class II molecule. And it's going to recognize that with the CD4 protein. So this recognition of antigen presented in an MHC2 molecule is the first activation step. So first, we could say for antigen presentation, antigen is presented in the MHC2 molecule. Antigen presented in MHC2 is recognized by CD4, and that's our first activation step. Now, we're going to have some co-stimulatory molecule involved. It could be a protein on the surface of our antigen-presenting cell that binds to a protein on the surface of the T helper cell, or it could be a co-stimulatory molecule that's released. Some second co-stimulation step is going to be required, and what's going to happen is my T helper cell is going to release interleukins that are going to come back and bind on it in an autocrine fashion. So this is the other type of cell signaling we have, autocrine. It's a message to self. And so what we could say happens for our second activation step is that the T helper cell releases interleukins that bind in an autocrine fashion, and this is our second activation step and now I'm activated. To do what? Form a clone. So now I'm activated to form a clone. A clone of what? A clone is now a large group of cells activated to this specific antigen. Okay? So that's the fourth step of a T helper cell's life cycle. The fifth step is going to be its proliferation and differentiation. So this proliferation is the formation of this clone. So I had antigen encounter and activation, and then I have that activation is going to lead me to form a clone of cells. So we can think about this as proliferation. So we'll proliferate into a clone of cells, which is a large group of cells that are activated to this specific antigen. And then I'm going to differentiate into effector helper T cells and memory helper T cells. My effector helper T cells are going to carry out my helper T cell effector response, which is to pump out interleukins. These interleukins are going to help activate our other lymphocytes. The memory T helper cells are going to hang out in secondary structures and await future encounter. Okay. Now what about our cytotoxic T cells? Alright, they originate in bone marrow, they go out to the thymus, undergo positive and negative selection, and they maintain their CD8 protein. And then they go off and seed in secondary lymphatic structures and await encounter. So how do they get their antigen encounter? Well, this time our cytotoxic T cells are going to, remember, have our CD8 protein, and now we're going to have some either antigen presenting cell or a sick cell that's going to have this antigen presented in our MHC class I molecule. So this is the first step in antigen encounter. So a cytotoxic T cell encounters its pathogen, or encounters its antigen, presented in an MHC1. Now, if this is all that happens, then these cells will just stay stuck that way. Because as soon as our cytotoxic T cell becomes activated, it's going to form a clone of cells that are just going to go start killing our own cells. And we don't want that to happen unless we're really sick. So the co-stimulation in the second stimulation step is really important for these cells, and that's why helper T cells are required. The effector response of our helper T cells is to pump out interleukins and help. Those interleukins are the second activation step for our cytotoxic T cells. So our first activation step is when we encounter antigen presented in MHC1. And that's all that's going to happen. In order to actually be activated, we have to have our second activation step, which comes when interleukins from an activated helper T cell bind to my cytotoxic T cell. So I have to already have activated a helper T cell. So let's say this helper T cell is hanging out in a lymph node and it's doing its thing, which is to pump out interleukins. So I'm a helper T cell, I was activated, I formed a clone, so there are actually a bunch of helper T cells here. We're in a lymph node and we're all doing our effector response, which is to pump out interleukins. Okay, well, interleukins then are going up, and now these interleukins can bind to my cytotoxic T cell, and it's activated. Activated to do what? Form a clone. So our first antigen encounter step happens when we're presented antigen in MHC1. The second activation step happens when interleukins from an already activated helper T cell bind to the cytotoxic T cell, and now it will be activated to form a clone. So I'm going to form a clone. Okay, so that's my proliferation and differentiation. So what does that look like for my cytotoxic T cells? I'm going to proliferate into a clone of cells that are ready to do their thing. I'm going to differentiate into the cells that are going to do that thing. What are they? My effector cells, which are my cytotoxic T cells, and my memory cytotoxic T cells. So I differentiate into effector cytotoxic T cells and memory cytotoxic T cells. Cytotoxic memory T cells hang out in secondary lymphatic structures and await encounter with the pathogen in the future. The effector T cells go out and carry out the effector response, which is to induce apoptosis in cells presenting this antigen in their MHC1. And they're going to induce apoptosis the same way that our natural killer cells did. So let's say this is blue cold virus, and here's my sick cell. He's presenting blue cold virus in MHC1. Now all of my effector cytotoxic T cells are not going to require the 1-2 activation step. They're effectors now that formed out of that activated clone. So their CD8 protein is going to recognize blue cold virus and immediately induce apoptosis by releasing perforins, which are going to perforate the membrane, and injecting granzymes so that my cell shrivels up and dies. The humoral immune response is the B cell response, and it's carried out through antibodies that are floating around in your blood or your body's humors. So our origin of B cells is in the red bone marrow. They mature in the red bone marrow. They leave red bone marrow naive but immunocompetent and hang out in secondary lymphatic structures awaiting encounter with their antigen. So how do B cells encounter their antigen? B cells fight extracellular pathogens. So their activation is slightly different than T cells. So our B cells have B cell receptors that kind of look something like this, and this actually looks a epitopes. So in order for a B cell to be activated, several epitopes have to bind lots of B cell receptors, and it's going to stimulate what's called cross-linking of the receptors inside the cell, and then will induce receptor-mediated endocytosis, where all of those bound receptors come into the B cell, and it can process the antigen and present it. So what's going to happen in our B cells, we'll say this is a bacteria, and all these little blue blobs are the epitopes, then our B cell receptors are going to bind to several epitopes of this antigen, and that's going to induce receptor cross-linking. So that's going to be something that's really important. So for antigen encounter, what we can say is that several B cell receptors bind antigen and cross-link. What does that mean? Well, inside the cell we'll have all of these changes that occur, so these receptors cross-link, and now this is going to induce in receptor-mediated endocytosis. So what does this look like? Well, if we were to zoom into the B cell, it's going to pull in this vesicle with all of those bound receptors. So that what happens is it pulls in this whole group of plasma membrane and all of the bound receptors, and now it can process that and become an antigen presenting cell. So after this we could say that so like maybe for a third step, the B cell processes the antigen and presents in MHC2 because it's an antigen presenting cell. Who's going to bind MHC2? CD4. CD4 is on our helper T cells. So guess what? We have to have a helper T cell activated to this pathogen. I'm not going to go over the helper T cell response again, but let's say that what we're fighting now is blue bacteria. In order to get past this step, we already have to have a T helper cell who's been activated to blue bacteria. So that activated T helper cell is sitting in a secondary lymphatic structure, pumping out interleukins. And now our B cell ingests and processes blue bacteria and is going to present blue bacteria in its MHC2. An activated T helper cell to blue bacteria can come along, and now we'll bind our interleukins, and now we can become activated to form a clone of effector cells. So our third step is that our B cell processes the antigen and presents it in MHC2. Next, we could say our activated helper T cell binds, and then we'll put in parentheses with it CD4. And then we could say that it's going to, our helper T cell releases interleukins that bind to and activate our B cell. What is our B cell going to be activated to do? Form a clone. So I get activated to form a clone. What's a clone? Well, it's this large group of cells that are going to proliferate and be already ready to fight blue bacteria. So I form a clone, and I'm going to differentiate now again into my effector cells and my memory cells, but my effector cells are different this time. So I form a clone. Again, this is a large number of cells activated to the specific extracellular pathogen. B-cell response. In the B-cell response, my effector cells are called plasma cells, and they release antibodies that are specific to this extracellular pathogen. Antibodies mark the pathogen for removal from the system in some other way that we'll talk about in just a minute. So our effector cells are plasma cells, and they release antibodies specific to this antigen. And this is an extracellular antigen. So I'm actually going to zoom ahead real quick to your antibody slide. We're not going to worry about the structure of an antibody too much. You can talk about that in an immunology class. But they kind of have these Y-shaped structures. And all of this part is like fixed. It's the same on all of our antibodies. What changes are these variable regions on the end. And those variable regions, these get programmed by the B-cell receptor. When a B-cell is specifying to an antigen, those variable regions are getting programmed to the specific pathogen that's in the body, or the specific antigen that's in the body. So now, our antibodies have these ends that look like that variable region that can bind to the epitope on the antigen, and then this fixed region can be recognized by some other part of the immune system, or can be removed from the immune system in some way. So the structure of our antibodies is that they have these variable regions that get specified to the specific epitope we're fighting. And then they have these fixed regions that are the same and are easy to recognize. So we'll say easily removed, or helps for the easy removal from the system. I'm just not going to say anything to cut right there. Okay, so what do antibodies do? Antibodies mark the antigen for removal from the system somehow. What are the ways that we can do this? Well, there's got to be a poem in here somewhere. Maybe you can write it for me. They do neutralization, agglutination, precipitation, complement fixation, and enhanced oxidation. So the variable region of our antibody is going to bind to the epitope on the specific pathogen, and then we're going to be able to do different things using that fixed region in different ways. So neutralization, when you make something neutral, it can't have an effect. So what happens in neutralization is that you bind all epitopes, so the antigen can't interact with your cells. So if this is a bacteria, and these are the epitopes, by sticking an antibody on here, I can just block all the epitopes, and if it can't bind with my cells, it can't do anything. In agglutination, I can stick all of my fixed regions together, clump them together, and then filter them out in some area like the spleen. So in agglutination, I am clumping together large particles. So this clumps together large particles. So I say I'm down to something here, and then I have another one here, and another one here. I can stick all of my fixed regions together and clump together large antigens. Precipitation is like agglutination, but for small things like toxins. So like you've heard of the tetanus shot, well that's to give you immunity against the tetanus toxin. So precipitation is when we use an antibody to stick to something small like a toxin and then clump it together. So precipitation clumps together small particles. And then finally, we can use this fixed region to stick a complement protein to an antibody. And so that will enhance everything that complement proteins do, like opsonization, flagging for removal by a phagocyte, enhance inflammation, or enhance cell lysis. So complement fixation, we could say that we can bind complement proteins to the fixed region of an antibody and enhance everything that complement proteins do. Okay, so that's the B cell response. The slides that I skipped over, let's come back and look at. So it's hard to measure the number of effector T cells that go out in the body. So it's kind of hard to measure the effect of a cell-mediated immune response. It's not hard to measure antibodies. So what we can do to kind of understand how different immune responses work is that we can look at plasma concentration of antibodies to particular extracellular pathogens. So in a primary response, that's the first time you encounter a specific pathogen, and that's going to require your two-step activation and your proliferation and differentiation and all of that. In your secondary response, your memory cells are going to be immediately activated to form a clone of new cells, and they will mount an immune response so quickly and so strongly that in theory, you should not even feel sick at all. So that's what we're talking about now. So memory is what we get. Memory is what gives us immunity. And in our primary response, this is when we're forming our first memory cells to a specific pathogen. So in a primary response, this is the first time you encounter a pathogen. And if we were looking at a graph, and let's say this is the day that you are exposed to a pathogen, for the primary, you would start feeling sick. And let's say, I think it's about 5 days. In the first 3 to 5 days, you'll start seeing antibody level go up. So this is plasma antibody. And so you feel sick this whole time. I encounter it here. I'm feeling sick. On days 3 to 5, somewhere in there, my antibodies start going up. And after a little bit, I start feeling better. Antibodies go down. I'm good. So the primary response is the first time I encounter a pathogen, you get sick, and you form memory cells. Okay, this was blue bacteria we were talking about. Alright, so let's say, like, 3 weeks later, I get blue bacteria again. Now I'm going to have a secondary response. And a secondary response is every consecutive encounter with blue bacteria, or every time you meet the same pathogen in the future, even if it's the 5th time you've seen blue bacteria, it's still a secondary response. So secondary response, this occurs with every consecutive encounter with the specific antigen. And what's cool about secondary responses is that they are faster and stronger. So again, here's our days, here's our plasma level of antibodies. So I'm just going to draw them. So now let's say we're feeling better, and here we get exposed to blue bacteria again. Well, in our secondary response, we're like immediately going to produce a bunch of antibodies, way more and way faster than the first time I had blue bacteria. So this time I don't get sick. Alright, well let's say green bacteria comes along. Does my immunity to blue bacteria protect me at all from green bacteria? The answer is no. So if I encounter green bacteria here, I'm going to get sick again. And after a couple days, I'll start seeing my antibodies to green bacteria go up. So your immunity to one thing does not give you immunity to another thing. Okay. So the last thing then that we're going to talk about is different ways that we can achieve immunity, and which is protection from disease, right? So we can get it actively or we can get it passively, and then in either way, actively or passively, we can also get it naturally or artificially. So what are we talking about here? In active immunity, I am actively forming lymphocytes to an antigen. So in active immunity, I actively form lymphocytes to a specific antigen. That's what makes it active, is that I'm actively forming this clone of specific lymphocytes. And this can happen naturally when I encounter the pathogen out in the environment. So if you're on spring break and the person that you're partying with has COVID and they rub all over your face, that's natural active encounter, and it should give you natural active immunity. So natural, you encounter the antigen out in nature or in the environment. Encounter the antigen in the environment. In the external environment. Or the natural environment. Because the doctor's office is also part of the environment, and that's where you're going for artificial immunity. Vaccines give you artificial active immunity. So what you do is you give like an innocuous version or sleeping version or dead version of some antigen to your body, and it still has that epitope that can be recognized by your lymphocytes. So it's active immunity because you're still actively making lymphocytes, but it's artificial because it's synthetic. The means of your exposure is synthetic. You're artificially being exposed to this pathogen. So that's what vaccines are doing. So you encounter the antigen synthetically. So this is like a vaccine for your flu shot. We're working on the COVID vaccine right now. Okay. The other way you can get some protection is passively. And this is passive because your body isn't doing anything to give you the protection that you're getting at the moment. So if you get bit by a black widow, you don't have long enough to mount a primary immune response and make anti-black widow toxin antibodies before you have neurological damage. So you need to race to the emergency room and get antitoxin right now. So that's passive immunity because you're getting protection. All that antitoxin that they're going to give you is going to bind up all the black widow venom in your system and you'll be protected, but it was passive because you didn't do anything in your body. I mean, you did have to get to the emergency room, but your body didn't actively produce anything. So passive immunity is your body is getting the protection from somewhere else and it really only lasts as long as this external source is present. So it's not long lasting, it only lasts as long as this is there. So only short-lived. So a natural example of passive immunity is the kind of immunity a baby gets from the placenta, passing mom's antibodies through to the baby, and then also babies passively get immunity when they're nursing. So breast milk contains antibodies that mom's producing, and then we'll give babies some passive immunity. So that's natural. So we could say antibodies in mothers give babies immunity or some protection. I'll say some protection. And this is both from the placenta in utero and from breast milk during lactation. So I once saw a cool Facebook post where this woman who must have been married to a scientist and was also breastfeeding her baby, showed these two different bags of her breast milk. And when a woman first delivers a baby, she's not going to produce milk right away. At first she produces this stuff called colostrum. It's like this straw-colored fluid that's high in antibodies. It gives that new baby some passive immunity right away. And it's yellow. It's like yellowish. And so after a couple days, when milk protein synthesis really kicks up, milk will be white. Well, this woman showed these two bags of her breast milk, and one of them was really yellow, like colostrum. So she or her scientist, whoever was the scientist, went and took it off and tested it, and what they found is that it more closely resembled colostrum. It was really high in antibodies, but it was high in antibodies to an infection that her baby was fighting, not her. So she wasn't sick at all. So what was really cool is that through the suckling that her baby was doing, it was exposing her body to the antigen, and her body made the immunity to it even though she didn't get sick. So that's one of the really cool protective things about breastfeeding. So that's a great example of natural passive immunity. And then the example that I talked about with the black widow bite, that's artificial. So in artificial passive immunity, what's happening is you have gotten bit by a rattlesnake or a black widow, and you've got this toxin in your body that's going to cause problems so fast that you call 911, you get in the ambulance, your friend takes you to the emergency room, whatever it is you got to do, and they give you antivenom. So what they do is they produce these antibodies in some other species, like a horse. So you can get the horse like over and over and over with rattlesnake venom. Not enough to kill the horse, but enough for it to start producing anti-rattlesnake venom antibodies. So then you can take those antibodies out of the horse's blood, and now when you race down to the hospital because you just got bit by a rattlesnake, they can give you these horse anti-rattlesnake venom antibodies to give you some protection. So all of those antibodies that you were given will bind up all of the rattlesnake venom in your body, and then once that gets cleared, you'll lose the protection. So artificial. This is when we could say antibodies are formed in another organism and given to provide quick immunity. And then we'll say, for example, horse antitoxins are used in humans with deadly bites. Okay. So there will always be an overlap. You can't just have passive immunity, and you can't just have active immunity. What type is it? Is it natural passive immunity, like the immunity a baby's getting from its mom? Is it artificial passive immunity, like the immunity you get if you need an antitoxin? Is it natural immunity? Is it active? Am I actively forming, is it natural that I'm actively forming lymphocytes because somebody sneezed on me? Or is it synthetic, that I'm forming lymphocytes because I went down and got my yearly flu shot? So just remember that. And that wraps up our discussion of the immune system. Stay healthy, be well.